Hey everybody, and welcome back. Here we are looking at week 7 of Geology 309, Bringing Fossils to Life. I've decided to take this week's lecture content and break it into 2 parts. So rather than have a very long lecture video, I'm breaking it into 2 lecture videos. This first part will deal with phylum Archaeocyatha, phylum Porifera, and phylum Cnidaria. We're now getting into the point of the class where our units are going to be looking at the individual phyla of organisms on the planet and looking at some of their main characteristics. So to keep things a little bit more organized, we're going to start with part 1, and when you're finished with this one, you can move on to part 2. So let's jump right into part 1. So these are our condo living organisms, more or less. We're looking at Phyla Archaeocyatha, Porifera, and Cnidaria, and these are all colonial organisms, or most of them are colonial organisms. So archaeocyathans are an extinct group of organisms we'll talk about here in a moment, but Porifera are sponges, hence why I have SpongeBob on here. And Cnidarians include corals, but also things like sea anemones and jellyfish. So though the other 2 organisms featured on this image, including a mammal and an echinoderm we'll be covering later in the semester, we're going to start with archaeocyathans, poriferates, and Cnidarians. To start out, let's look at phylum Archaeocyatha for just a brief moment. Now, this is a phylum of organisms that is entirely extinct today. These were around exclusively from the early through middle Cambrian. However, they're very important as being the first metazoan reef builders. So the word metazoan, if you're not familiar, means multicellular organism. Easier to say metazoan. And they're reef builders in the early Cambrian. So these were the dominant organisms that were building some kind of structure that other organisms would live around. They are passive filter feeders that are composed of an inner wall and an outer wall with structural septa kind of holding them together. They had little roots that would attach them down into the sediment and this very porous body. Like sponges, they were probably made of a series of semi-independent cells, but they're a passive filter feeder. So in other words, they would get food or nutrients by just sitting in the water and letting water wash through them. And then these semi-independent cells that are lining the body wall would then absorb those nutrients. So they actually don't do anything. They just sit there in the water waiting for organic material to filter through them, and they digest it. You're probably thinking they aren't going to get a lot of nutrients doing this way, and you're right. But then again, if you don't do anything, if you're not physically getting up and moving around, you don't have much in terms of metabolic needs anyway. So these are one of the earliest colonial, I guess, reef builders, and they would have needed some kind of a constant current flow. What's interesting is that inner wall and the outer wall, and you've got those compartments in between, that's going to actually slow water down as it's flowing through the archaeocyathon, allowing those bits of organic debris to fall out of suspension and thus become digested a little bit easier. So these are an animal. As you can see, they're very small. In cross-section, they have you can see the radiating septa. But in life, they were kind of cone-shaped. So we're going to talk about these for a minute, and now we're going to move into sponges, which is kind of like the next stage in archaeocyathans. Phylum Porifera is essentially what we find with sponges. Okay, a lot of our sponges, though, in the fossil record, break apart into very small pieces, so they're studied as microfossils. Now, these individual pieces we call spicules. And spicules can be made of a variety of different things. They can be made of silica, they can be made of calcium carbonate, and in some cases they can be organic. So if you find a sponge on the beach and you can squeeze it and it's spongy, that's an organic spicule, or it's made of organic spicules. However, our glass or siliceous sponges, not something you'd want to use for cleaning yourself. That would kind of hurt. And the calcareous ones actually hold up quite well in the fossil record. But when a sponge disaggregates, it produces all the little needle-like structures that you see before you. So if we want to look properly at phylum Porifera, we know that sponges are around in the Cambrian, possibly Precambrian, but definitely in the Cambrian and of course still around today. Again, a sponge is actually a colony. So when you look at any of the sponge images on the screen, these are colonies of semi-independent cells. In other words, each cell is its own individual. living in the colony. However, it can't function exclusively on its own. It needs the colony to survive, hence why we say semi-independent. So when the living cells are then broken off, they can regenerate into a new individual if you have enough cells to form and start a new colony. Sponges can be in a wide variety of different shapes and sizes. We see large basket-shaped or tube-shaped, sometimes cones, and sometimes they're just small and kind of encrusting, formless sponge-type organisms. Now, unlike archaeocyathans, which kind of passively sit there and let water flow through them, sponges actually do pump water, except they do it with flagella. Some of the cells have little hair-like structures, and they will whip those back and forth, and they're microscopic. And so this microscopic whipping motion of these little tiny flagella will kind of waft water towards the sponge and through the sponge. So they're doing a little bit more motion than archaeocyathans, but not much. Again, they're not absorbing a ridiculous amount of nutrients here, but again, they don't really need it. So the flagella are on that outer wall covering, which we call, into a central canal. The center of the sponge is known as the osculum. And so the flagella are pulling water and pumping water kind of into that osculum where they'll filter out any organic material, and then they pump water out the other side. As we mentioned, the skeleton or the main structure of a sponge is made of spicules that can be composed of silica, calcite, or calcium carbonate, or an organic compound. And that's one of the ways in which we try to phylogenetically classify sponges into different classes. So there are 4 main classes, but right away we're going to notice that we have some problems with this classification. Class Hexactinellida. These are all of your siliceous sponges, so the glass sponges. Class Calcarea are obviously calcareous spiculed sponges. Class Demospongia includes a combination of spicules made of spongin, which is an organic compound, and or silica. So these are ones that might have silica. They're a little different than the hexactinellids. We're going to look at some examples here in a minute. And then we have class Sclerospongia. Now, we don't have many sclerosponges alive today. It does include the extinct sponges, the stromatoporoids, which were huge reef builders in the Silurian and the Devonian, so kind of a cross between a sponge and a stromatolite. Today, we actually consider all of the fossil sclerosponges we find to be polyphyletic. They have characteristics that appear in all sorts of different groups. Still, because that early evolution of sponges is not fully resolved, we keep the polyphyletic clade, recognizing though that it is probably false, but there might be some members that belong in it together and others not. So this is what we do when we have a polyphyletic situation, is we know that the tree needs some work, pruning for instance, but we're going to maintain it as we collect more data on more fossil sponges. Now, in the late Devonian, especially towards the end of the Frasnian-Famennian extinction event, reefs were predominantly formed from sponges. As we lost most of our Paleozoic corals in the mid-Paleozoic, reefs were then composed of sponges, specifically this hexactinellid sponge Hydenoceras. It's a very famous siliceous sponge, and the very first to colonize right after those corals went extinct. So here we can see our so-called sclerospongid stromatoporoids, which again are from the Cambrian to the Jurassic, though again, because we're looking at a polyphyletic group, maybe they're still around today. Maybe that's what some of our modern sponges derive from. We're not really sure. But again, you can see that there is a definitive structure to these. Look at the dimpling pattern on the surface. And then in cross-section, you can see the individual layers. So these really are kind of a cross between a stromatolite and a sponge. Essentially, it's a sponge that grows very thin layers, similar to the way a stromatolite would, accidentally accumulate sediment while these microbial mats would be forming. In this case, it's semi-independent colonial cells. And I briefly want to bring up receptaculites, which is very abundant in Ordovician rocks, but we have them from the Ordovician through the Triassic. These are actually not sponges. They look like sponges, and they were long thought to be sponges, but receptaculites are actually an algal mound. But again, a very, very common fossil to find even in some of our local rocks, but not a true porphyrid sponge. And now this brings us to phylum Cnidaria, which include a wide variety of some of our dominant reef-building organisms in our oceans today, and going all the way back to the Precambrian. So cnidarians all have tentacles with stinging cells that we call nematocysts. This is true for corals, sea anemones, and for jellyfish. We have three classes of cnidarians. The most important one that we're going to be talking about is class Anthozoa, because these make up more of our fossils than the other two classes of cnidarians. Anthozoan cnidarians include corals and sea anemones. We have two subclasses of Anthozoa. Octocoralia. Now these are going to have 8 tentacles per polyp, which is what the actual animal in a cnidarian looks like. This includes our soft corals, our sea fans, sea whips, sea pens, and then subclass Zoantheria. These have tentacles in multiples of 6, so 6, 12, 18, 36, so on. These are our hexacorals. These include 3 orders: tabulates, rugosins, and our modern-day stony corals, the scleractinians. So, just to do a quick recap so far on Anthozoa, we have Octocoralia. These are soft corals. And then we have Zoantheria, and these are your stony hexacorals. So if you're looking at fossil corals, we're mostly focusing on Zoantheria. Of the orders of Zoantherian Anthozoan cnidarians, we have tabulates and rugosins, which are Paleozoic, and our scleractinians, which first appear in the Triassic and are still around today. Our other 2 classes of cnidarians include the hydrozoas, or hydrozoans, and scyphozoas, which are the more medusoid jellyfish, the ones that float around. So let's look at these from the terms of the fossil record and what we can learn about them by looking at modern-day cnidarians. Now, most of your anthozoan corals, the animal itself is in the form of what we call a polyp. It's a small kind of barrel-bodied organism with a mouth that's surrounded by those stinging tentacles. And inside of the body cavity, we have a coelenteron lined with mesenteries, essentially digestive tissues. Now, if you look at a medusoid cnidarian, so something like a jellyfish, they're actually very similar, just kind of flipped upside down. You still have tentacles, you still have that are ringing around a mouth, and you have this body cavity of a coelenteron. So they're very similar. In fact, some of our organisms here, at least on the polyp side, start out in a medusoid form and then during their ontogeny convert to a more stationary polyp. Our Paleozoic corals are, again, quite different than our modern day ones. And most of our huge Paleozoic reefs were actually in the Devonian. That's when we had the most diversity of reef-building organisms, especially corals, where we had the tabulates, the rugosins, and then we also had quite a lot of stromatoporoids as well. Hopefully you all recall that we have 2 orders of our Paleozoic corals. The first order we're going to look at is order Rugosa. Now these can be solitary horn corals, such as Streptolasma, or colonial, such as Hexagonaria. The order Tabulata, the other Paleozoic coral order, are all colonial, whether we're looking at Halocites or Favocites or many others. These are all colonial, and rugose ones have colonial members and solitary ones. I do want to point out that in our rugose corals, the polyps that make these stony coral structures are much larger than the colonial corals that make up Tabulata. So order Rugosa existed from the middle of the Ordovician all the way through the end of the Permian, though most of these corals had their greatest decline at the end of the Devonian. All rugose corals make an exoskeleton that is made out of calcium carbonate. And again, they can be solitary, such as we see with Streptolasma, or they can be colonial, like we see with Hexagonaria. One important thing to point out about rugose corals is how they form their radiating septa. So when you look inside the coral structure of a rugose coral, you'll see these little partition walls. Okay, these are known as septa. This is what the polyp uses to grip and attach itself to the inside of its exoskeleton. Now, the way that rugose corals grow their septa is through what we call serial insertion. So, for example, they will grow their first main septa, that would be the first generation, and then the second generation grow kind of oblique into the side, and then the third, and then the fourth, and notice that they just kind of grow around as they radiate towards that center. Okay, so this is a very unique way of growing septa compared to some of our more modern corals. During the Devonian period, when we had these massive reefs of both tabulate and rugose corals, we actually find that rugose corals would grow daily and annual growth layers and bands. So when we look at the individual layers, each one of those was produced in roughly a one-day time frame. And then we have major bands that indicate annual growth layers. Now, how do we differentiate an annual versus a daily? Think about today, if we were to look at tree rings, you'd be able to tell changes in seasons, changes in precipitation, changes in temperature. And so we can actually see how many daily layers we have in an annual band. What we have discovered with rugose corals, if you look at the average of these rugose corals from the Devonian, there's an average of 405 layers within 2 annual bands, so 405 layers a year. We would expect modern corals today that might be putting down annual layer or daily layers to have 365 in a one-year period. But with 405 layers, that means we have more days, essentially, in a year. So what does that actually mean? It means that the Earth was spinning faster back in the Devonian than it is today. Order Tabulata, we do have them in the Cambrian, but they're quite rare. We know we have them in the early Ordovician, and again, through the Permian, and just like the rugose, we lose most of them at the end of the Devonian. Okay. All tabulate corals are colonial, and the reason they're called tabulates is because each coral colony is composed of numerous little tablet-like components. So if you were to take some of the more common tabulate corals like Favosites or Halosites and cut them long ways, you'll see a series of little boxes. Each one of these housed a polyp, and each box has a little window that the polyp could reach out and feed by zapping whatever detritus fell by and then pulling it into the mouth. But then when they were in a dormant stage, they would pull themselves back inside of their little apartments. So these are all essentially like condos, and they're all living right next to and on top of each other. Favosites is a very well-known tabulate coral. When you look at it straight down, you can see this kind of six-sided structure making up our hexacoral. But when you look at it from a side cross-section, you can see all of the little tabulae, the little floors and ceilings making up those individual compartments. This is sometimes called the honeycomb coral. Halosites is another very common one. Again, we have tabulae, and again, we have these little 5 to 6-sided structures, but in this case, the individual coral structures grow in almost what looks like a chain link pattern. This dark space you see here, that's not where a coral is living. The corals are living in the individual little tiny holes you see making up our so-called chain link walls. But again, in cross-section, lots and lots of tabulae, little floors and ceilings. Our modern hexacorals are all members of order Scleractinia. These appear in the middle Triassic and are still around today. So if you go snorkeling or scuba diving and you see corals, you're looking at Scleractinians. Most of these are colonial. We do have some solitary Scleractinians, and they actually make a calcium carbonate structure that's truly aragonitic rather than calcitic. They are hermatypic reef formers. What's interesting about our modern day reef forming scleractinians is that they have a symbiotic relationship with a type of algae known as zooxanthellae. Zooxanthellae algae is photosynthetic, like all algae. So the corals actually need to live in relatively shallow water where they can get solar radiation to feed the zooxanthellae algae that are living inside of their cells. These algae are protected by the nematocysts, the stinging cells of the coral itself, and they provide the coral with a significant portion of its food source. So if you actually were to block out the sun from some of these corals, they would die, but they wouldn't die because they don't get solar radiation. They would die because their zooxanthellae wouldn't be getting solar radiation and wouldn't be producing food, and thus the corals would die from a slow starvation. We also do have some ahermatypic non-reef forming that don't have these symbiotes, and they live in deeper water facies. So we do have some types of corals that don't need to be near the sun or need to be near the surface, but a lot of them do. So if you're a reef-forming coral today, they have this zooxanthellae algae living inside of their cells of the polyps. And when we compare our rugose corals to our scleractinians, we see both of them have radiating septa. But we talked about how the rugosans had a serial septal insertion. Scleractinians use what's called radial insertion, where they grow their main primary septa, and then in between those, we'll divide that with a secondary septa, and then a tertiary and a quaternary. So they're all going to ultimately meet right here in the middle, while the serial insertion grow into the sides of the previous generation of septa. So that's a quick way of identifying the difference between a scleractinian and a rugosan. So here's a couple of questions to consider as you're reviewing this video and as you're doing some reading. Did Paleozoic corals, like the rugose and the tabulates, have symbiotes like zooxanthellae that our modern ones do? Well, how would we test this hypothesis? First of all, what we'd want to do is look at what types of marine facies we find Paleozoic corals in. And sure enough, we find a majority of our reef-forming Paleozoic corals in relatively shallow marine settings. Now, we know that today we have the ahermatypic non-reef-forming corals that don't need to live in shallow marine facies, and they don't. They live in more deep water environments. So does the presence of Paleozoic corals in shallow facies indicate that they needed to be in sunlight because of some kind of symbiotic relationship with something like zooxanthellae? That's very likely. And where did scleractinians come from? Think about it. We lose most of our rugosans and tabulates at the end of the Devonian. Not entirely, but most of them. And we really don't have much for corals throughout the Mississippian, Pennsylvanian, or Permian. And it's not until the Middle Triassic that we start getting our scleractinians. So where did scleractinians come from? This coral gap, there's two different schools of thought on it. First is that we have the survival of rugoses with some kind of modification. Rugosans did make it all the way to the Permian. They were just very, very rare, and perhaps there was an off branch of those populations in the early Triassic that made it through the Permo-Triassic extinction that led to what we now call modern day scleractinians. The other possibility is a modification of a sea anemone. If you look at the polyp of a scleractinian coral and a sea anemone, they look very, very similar. The only difference really is the size. So those are some possibilities on where we actually get scleractinians. So far we've looked at metazoans. We're looking at multicellular life. And we see that we have two different, a couple of major groups of poriferates, but porifera as a whole is kind of problematic. We know that hexactinellids and our demosponges are closely related, and the calcareans are a little different. We're not really sure what's going on with archaeocyathans. They fit somewhere there. This is what we mean by a polyphyletic relationship with poriferids. We know something's wrong here, but this is still a work in progress. As we move up our phylogenetic tree, we start seeing organisms that have biradial symmetry. This includes cnidarians. Now, the relationship between tabulate, scleractinians, and rugosans, or our anthozoans, is still not fully understood, but we know that they group together. Cnidarians also are one of the first groups of organisms that are coelomates, that have an internal body cavity, and that's going to include everything up the tree from here. However, while Cnidarians have biradial symmetry, what we're now going to be getting into in part 2 of this lecture series, as well as the rest of the semester, is looking at organisms that have bilateral symmetry, so a mirror-like symmetry. And this includes our 2 main groups of multicellular life after Cnidarians, the deuterostomes and the proterostomes. And we will pick up with that in part 2.